Hello guys, welcome back to Medico Days. So today in this video, we'll be starting our new series of Inicet PYQ. So in this video, we'll be discussing about the November 2024 and July 2025 PYQ of one particular subject. But before moving into the subject, let's have a look at the subject-wise question weightage. So General Medicine July 2025 had 22 questions and November 2024 had 23. Physiology 15 and 20 respectively. Surgery 17 and 18. Micro, OBG. So I've kept it in the decreasing order. So you can go through this, guys, and some of the certain subjects like biochemistry, FMT, they also have quite good number of questions. They had quite good number of questions in July 2025, but there were only 6 questions in November 2024. So certain alterations are there. So this is the subject-wise weightage. And I have made a graph also, guys, about the May 2025 and November 2024 exam subject-wise Inicet weightage. Now moving on to the biochemistry questions of July 2025. An obese individual with a weight of 90 kg takes a high carbohydrate diet. Which of the below are most responsible for weight gain? So answer is 1, 2 and 4, that is malonyl-CoA, acetyl-CoA carboxylase and tricarboxylate transporter. So malonyl-CoA is a key intermediate in fatty acid synthesis derived from acetyl-CoA carboxylation. Acetyl-CoA carboxylase is a rate-limiting enzyme that converts the acetyl-CoA to malonyl-CoA, promoting lipogenesis. Tricarboxylate transporter, it facilitates the citrate export from mitochondria to cytoplasm, where it provides acetyl-CoA for fatty acid synthesis. Mitochondrial carnitine transporter is involved in fatty acid oxidation, that is breakdown, and not synthesis, and is suppressed during lipogenesis. Next, in the glycine titration curve, find the zwitterionic pH. So pK1 is 2.4 and pK2 is 9.8. Here the answer is 6. So the zwitterionic pH, that is the isoelectric point for a neutral amino acid like glycine, is the average of its two pK values. So that will become 6.1, and which is nearer to the value 6. Next, how many cycles of PCR is required to generate half the copies of 26 cycle? Answer is 25, because in PCR, DNA amplification is exponential, with the number of copies doubling each cycle. So after n cycle, the copies will be 2 raised to n. So after 26 cycle, the copies will be 2 raised to 26. Half of this is 2 raised to 25, which is achieved after 25 cycles. A 4-year-old child presents with developmental regression, hepatomegaly and cherry red spot in the macula. The startle reflex is normal. Which of the following lysosomal enzymes is deficient? Answer is sphingomyelinase. The clinical features: developmental regression, hepatomegaly, macular cherry red spot, and a normal startle reflex. They are all classic for Niemann-Pick disease type A. It results from a deficiency in acid sphingomyelinase, leading to the accumulation of sphingomyelin in macrophages and the central nervous system. So others like hexosaminidase A deficiency, which causes the Tay-Sachs disease. It also causes neurodegeneration and a cherry red spot, but features of exaggerated startle reflex, and there is no organomegaly seen in them. And next, glucocerebrosidase deficiency, Gaucher's disease. It causes hepatosplenomegaly and hematological abnormalities, but no cherry red spot or rapid neurodegeneration is seen there. Next, aryl sulfatase A deficiency, that is metachromatic leukodystrophy. It primarily causes demyelination and motor regression, but there is no cherry red spot or hepatomegaly seen. Next, a neonate was normal at birth but developed liver disease and muscle weakness 4 months later. She had severe hypoglycemia, particularly in the morning after an overnight fast. On examination, there is hepatomegaly. Lab test done after fasting shows ketoacidosis. pH is 7.25 and increased ALT and AST levels are seen. On administering IV glucagon after a carbohydrate meal, a normal rise in blood glucose levels is seen, whereas on giving IV glucagon after an overnight fast, there is no rise in blood glucose. Liver biopsy shows increased glycogen, 8% of the weight. Which enzyme has a defect? Answer is debranching enzyme. So the clinical presentation of hypoglycemia after fasting, hepatomegaly, ketoacidosis, elevated transaminases, muscle weakness and glycogen accumulation, 8% of the liver weight, is all characteristic of glycogen storage disease type 3, that is the Core's disease. The glucagon test shows a normal glucose rise after a meal but no rise after fasting, pinpointing a debranching enzyme defect. This enzyme is essential for removing the branch and it branch points during the glycogenolysis. Next, the glucose 6-phosphatase deficiency, which is a Von-Jurke's disease. It would show no glucose response to glucagon in either state because of the blocked final step of glucose production. Liver glycogen phosphorylase deficiency, which is seen in Hirsch's disease. It causes fasting hypoglycemia and hepatomegaly, but typically lacks muscle involvement and shows a subdued glucagon response post-meal. Muscle glycogen phosphorylase deficiency, which is seen in McArdle's disease, it affects the skeletal muscle exercise tolerance, not liver function or fasting hypoglycemia. Next, cancer cells consume more glucose by which of the following process? Answer is increased lactate is produced even in the presence of oxygen. So cancer cells, they exhibit the Warburg effect, which is characterized by aerobic glycolysis with high glucose consumption and lactate production even in the presence of oxygen. This metabolic shift supports the rapid cell proliferation by providing carbon intermediates for biosynthesis rather than maximizing the ATP yield via oxidative phosphorylation. Cancer cells, they often upregulate the GLUT1 and they prefer glycolysis over oxidative phosphorylation. Next question. In glucose 6-phosphate dehydrogenase deficiency, what will be the bilirubin finding? Answer is there will be normal direct bilirubin, indirect bilirubin will be increased, and urine urobilinogen will be increased. So glucose 6-phosphatase dehydrogenase deficiency causes hemolytic anemia during oxidative stress, leading to excessive red blood cell breakdown. Indirect bilirubin is increased because of due to the heightened catabolism, and direct, that is a conjugated bilirubin, it remains normal as liver conjugation function is intact, and urine urobilinogen is increased because intestinal processing of excess bilirubin generates more urobilinogen, which is absorbed and excreted in the urine. Next, gene pool refers to all the alleles in answer population. So the gene pool encompasses the complete set of alleles present across all breeding individuals within a population. It represents the collective genetic diversity available for inheritance and evolution, influenced by mechanisms like mutation, selection, and genetic drift. Next question, which of the following is correct? Answer is northern blotting for RNA, southern blotting for DNA, and western blotting for protein. So this is again a very important question, guys. You can remember like west for proteins pizza, and southern blot DNA the dosha, and northern RNA roti. Like North Indians like roti, South Indians like dosa, and West people like pizza. So all the first letters of each. Next, cyanide and carbon monoxide acts on which of the heme proteins? Answer, cytochrome C oxidase. Cyanide and carbon monoxide are potent inhibitors of cytochrome C oxidase, that is a complex 4, in the electron transport chain. They bind to the heme ion in the enzyme, blocking ion electron transfer to oxygen and halting the ATP production, leading to cellular hypoxia. Myoglobin binds oxygen but is not a primary target for these toxins. Catalase decomposes the hydrogen peroxide and is not inhibited by cyanide or carbon monoxide. NADH dehydrogenase, the complex 1, is affected by other inhibitors. Example, rotenone, but not by cyanide or carbon monoxide. So that's it, guys, for this video. In my next video, I'll be coming up with next set of questions. So till then, stay tuned, and if you have any other suggestions, please do mention it in the comment box. If you're interested, you can buy my NEET PG previous year questions Q bank. The link is there in the description. So that's it, guys. Till I come back with my new video, stay tuned. Till then, bye.